This video outlines the sequencing process undertaken during the making of creative intervention between the lines. DNA sequencing is the process of determining the order of nucleotides in DNA. For this project, the sequencing process was performed by Exeter Sequencing Service at the University of Exeter, using the Illumina NovaSeq 6000 sequencing machine. The synthetic DNA containing the original text document arrived from Twist Bioscience in powdered form inside sealed metal capsules. First, we centrifuged the capsules to ensure no DNA was lost when it was opened. After piercing the capsule, TE buffer was added to dissolve the DNA. This buffer contains elements which help to preserve the DNA. Once dissolved, we left the solution on ice before the amount of DNA present was quantified using a qubit device. Using a fluorescent dye that binds to the DNA, this machine can measure the sample to see how much has been recovered from the capsule. To increase the sample size, we then used an amplification process called PCR to make lots of copies of the DNA. The PCR cycle was repeated 9 times, amplifying the DNA by a factor of approximately 500. The DNA solution was then purified using SPRI beads, a buffer containing magnetic ferrous beads. The DNA binds to these beads and the remaining solution can be removed. The beads were then washed several times in 80% ethanol, which was then allowed to evaporate. A new buffer was then introduced for the DNA to dissolve into, unbinding it from the SPRI beads. To measure the sample quality, the DNA was then run through a tape station on screen tape, a small cassette with gel-filled capillaries. During this process, DNA is separated by the size of its base pairs and compared against a ladder of molecules with known sizes. The tape station provides a printout, which in this case indicated that the initial DNA's 196 base pairs had increased to 250. The extra base pairs showed that the additional DNA barcodes needed for sequencing were now present in the DNA. The DNA was then sequenced using the Illumina NovaSeq 6000 sequencing machine. The sequencing process identifies the sequence of the DNA nucleotides within a DNA molecule. Firstly, the DNA is denatured, turning it from a double-stranded helix to single-stranded molecules. The DNA is then added to a patterned flow cell to be analysed inside the machine. The sequencing machine determines the sequence by measuring and recording light signals emitted from the DNA nucleotides, with each base having its own unique signal. The outputted files detail the DNA sequence of the original DNA molecules. This sequence was then decoded back into binary data using Twist Bioscience's algorithm, and the text original document containing the students' stories and experiences was recovered with no data loss.